Hi, hello. What's up to all of you listeners and viewers out there? My name is Cromwell Cruz, and me and my friends JB and Lou are going to be discussing the molecular basis of inheritance. So, to start with it, let's discuss what is a DNA. DNA is the most celebrated molecule of our time, for it is the substance of inheritance. Mendel's heritable factor and Morgan's gene on chromosomes are in fact composed of DNA. Chemically speaking, your genetic endowment is the DNA you inherited from your parents. For further and formal introduction and discussion of our topic, my friend JB here will be discussing DNA replication.
This is JB from Biado. The relationship between structure and function, one of the themes of biology, is manifest in the double helix. The idea that there is specific pairing of nitrogenous bases in DNA was the flash of inspiration that led Watson and Crick to correct double helix. At the same time, they saw the functional significance of the base pairing rules. Well, during DNA replication, base pairing enables existing DNA strands to serve as templates for new complementary strands. When a cell copies a DNA molecule, each strand serves as a template or mold for ordering nucleotides into a new complementary strand. One at a time, nucleotides line up along the template strand according to the base pairing rules. The nucleotides are linked to form new strands. Where there was one double-stranded DNA molecule at the beginning of the process, there are now two, each an exact replica of the parent molecule. The copying mechanism is analogous to using a photographic negative to make a positive image, which can be in turn be used to make another negative, and so on. To get started with this discussion, there is what we call an origin of replication. The replication of a DNA molecule begins at a special site called origins of replication. Replication recognizes this sequence and attach to the DNA, separating the two strands and opening up a replication bubble. Replication of DNA then proceeds in both directions until the entire molecule is copied. In contrast to the bacterial chromosome, each eukaryotic chromosome has hundreds or thousands of replication origins. Multiple replication bubbles form and eventually fuse, thus speeding up the copying of the very long DNA molecules. A helicase is an enzyme that untwists the double helix at the replication fork, separating the two old strands. Molecules of single-strand binding protein then line up along the unpaired DNA strands, holding them apart while they serve as templates for the synthesis of new complementary strand.
Now, Lou will now discuss the lagging and leading strand.
Hi, I am Lou from Biado, and I will be discussing to you the leading and lagging strands of DNA. In the leading strand, a short piece of RNA called a primer, which is produced by an enzyme called primase, comes along and binds to the end of the leading strand. The primer acts as the starting point for DNA synthesis. DNA polymerase binds to the leading strand and then walks along it, adding new complementary nucleotide bases.
To further proceed DNA replication, elongation of a new DNA at the replication fork is catalyzed by enzymes called DNA polymerases. As nucleotides align with complementary bases along a template strand of DNA, they are added by polymerase, one by one, to the growing end of the new DNA strand. The rate of elongation is about 500 nucleotides per second in bacteria and 50 per second in human cells.
Okay, so priming DNA synthesis. There is another important restriction for DNA polymerases. they can add a nucleotide only to an existing polynucleotide that is already paired with the complementary strand. DNA polymerase cannot actually initiate synthesis of polynucleotide. They can only add nucleotide to the end of an already existing chain. In the cell, the original pre-existing chain, the primer, is not DNA, but a short stretch of RNA. Another DNA polymerase later replaces the RNA nucleotides of the primers with DNA versions. Only one primer is required for a DNA polymerase to begin synthesizing the leading strand of a new DNA. For the lagging strand, each fragment must be primed. The primers are converted to a DNA before ligase joins the fragments together. Okay, thank you very much guys for watching this video, and if you liked and enjoy it, hit that subscribe button below, and if you are interested in our other videos, go and subscribe to us in Facebook, YouTube, Instagram, and Vine.
That is bio for
Okay, so yeah, that is BioPro, and thank you for listening
Yay!
and watching.
Thank you for watching. I can.
Awesome, yeah. Awesome.